A bulldog struggles to breathe, not from exertion, just from existing. Its airways are so compressed that every breath is a fight for survival. This dog will live perhaps 7 years if lucky. Its ancestors lived 15. A German Shepherd's hips collapse at age 4. The joints that should last a lifetime have deteriorated into bone-on-bone agony. Surgery costs $10,000. Without it, the dog will drag its hindquarters by age 6. A Cavalier King Charles Spaniel screams in pain when touched. Its skull is too small for its brain, a condition called syringomelia. Fluid-filled cavities form in the spinal cord. There is no cure. The dog's own skull is killing it from the inside. These aren't isolated cases of bad luck. These aren't rare genetic accidents. These are the intended results of modern dog breeding. In 2021, geneticists at UC Davis completed the most comprehensive analysis of purebred dog genetics ever conducted. They examined the genomes of 49,000 dogs across 227 breeds. Their conclusion was devastating. Most modern dog breeds are more inbred than wild populations on the brink of extinction. The average purebred dog has the genetic diversity of a population of 10 to 50 individuals. Some popular breeds, beloved family companions, are genetically equivalent to populations of fewer than 10 individuals. We have taken wolves, one of the most genetically robust species on Earth, and reduced their descendants to genetic ruins. We didn't do this by accident. We did this deliberately, methodically, over 150 years in the name of perfection. Today, we journey through evidence that challenges everything we thought we knew about the dog. Prepare yourselves. We begin. In 2008, the BBC aired a documentary called Pedigree Dogs Exposed. It revealed that Crufts, the world's most prestigious dog show, was celebrating breeds with catastrophic health problems. Bulldogs that couldn't breathe, Cavaliers with skulls crushing their brains, German Shepherds with spines so deformed they walked like frogs. The outcry was immediate. The Kennel Club claimed ignorance. Breeders insisted these were isolated cases. But scientists had known for decades. They'd been publishing studies showing genetic collapse in purebred dogs since the 1980s. The evidence had been ignored. The story actually begins not in 2008, but in 1873, when the Kennel Club was founded in Britain to regulate dog breeding. For thousands of years, dogs had been bred for function: herding, hunting, guarding. Appearance mattered far less than ability. But Victorian England became obsessed with purity, standardization, and aesthetics. Dog shows became social events where wealthy owners competed for prestige. Breed standards were established, precise specifications for how each breed should look. Dogs were judged against these ideals like sculptures judged against blueprints. This created a problem. Achieving these standards required extreme inbreeding. To fix specific traits, a particular head shape, ear set, coat color, breeders needed to concentrate genes. They did this by mating closely related dogs, fathers to daughters, brothers to sisters, cousins to cousins, for generation after generation. The results were visually striking. Each breed became instantly recognizable, conforming precisely to its standard. But genetically, the consequences were catastrophic. Here's why. Every organism inherits 2 copies of each gene, one from each parent. Many genetic diseases are recessive, meaning they only cause problems when an individual inherits 2 copies of the defective gene. In genetically diverse populations, recessive diseases are rare. The odds of 2 carriers mating are low. But in inbred populations, those odds skyrocket. When dogs are bred to close relatives, both parents often carry the same recessive mutations. Their offspring inherit 2 copies and develop the disease. Imagine a genome as a library containing 20,000 books, genes. In a healthy, diverse population, each individual's library has perhaps 5 to 10 damaged books, but the corresponding books from the other parent are intact. Function is preserved. In inbred populations, both libraries have damage in the same books. No backup copy exists. The result? Catastrophic loss of function. By 2010, geneticists at the Broad Institute began sequencing purebred dog genomes to measure the damage. What they discovered was worse than anyone imagined. The coefficient of inbreeding, COI, measures how related an individual's parents are. A COI of 0% means no shared ancestry. A COI of 25% is equivalent to siblings mating. A COI of 50% is equivalent to parent-child mating. Conservative biologists consider a COI above 10% dangerous for wild populations. The Frozen Zoo in San Diego, which preserves genetic material from endangered species, refuses to breed animals with COI above 12.5%. They consider it genetic suicide. The average purebred dog has a COI between 20 to 40%. Some breeds exceed 50%. We are breeding dogs more inbred than species on the verge of extinction. But the COI only tells part of the story. The real damage lies in what geneticists call effective population size, the number of breeding individuals needed to maintain current genetic diversity. Dr. Danica Banish at UC Davis led a study calculating effective population size for 227 breeds. The results were staggering. Norwegian Lundehund, effective population of 8 individuals. English Bulldogs, effective population of 12 individuals. Flat-coated retrievers, effective population of 21 individuals. Bernese Mountain Dogs, effective population of 24 individuals. For context, conservative biologists consider an effective population below 50 a genetic emergency requiring immediate intervention. Below 20 is considered terminal. The species is doomed without dramatic genetic rescue. Over 75% of recognized breeds fall below this threshold. But here's what made researchers realize this was a crisis. These numbers represent the genetic diversity remaining today, after 150 years of intensive selection. Ancient dogs, even dogs from just 200 years ago, had vastly more diversity. When researchers sequenced Victorian-era dog remains and compared them to modern breeds supposedly descended from those dogs, they found modern breeds had lost 60 to 90% of their genetic diversity. Entire sections of the genome, bookshelves in our library metaphor, had been erased. But the evidence forces us to confront an uncomfortable possibility. The dogs we consider purebred aren't pure at all. They're genetic catastrophes, populations so inbred that most are less genetically diverse than lab mice bred specifically for genetic uniformity. The genetic collapse isn't theoretical. It manifests as an epidemic of breed-specific diseases affecting millions of dogs worldwide. In 1899, a German shepherd named Horrend von Grafrath was declared the breed standard. His descendants became the foundation of the entire breed. Today, 19% of German shepherds develop hip dysplasia, a crippling condition where hip joints deteriorate, causing chronic pain and lameness. But the numbers are deceptive. The true rate is far higher, approaching 50% in some lines, because many cases go undiagnosed until severe. The condition is so common that it's considered normal for the breed. Researchers at Cornell traced the genetic cause. A handful of genes affecting joint development became fixed in the population through inbreeding. Once widespread, these alleles became impossible to eliminate without crossing, introducing genes from other breeds, which breed standards forbid. Modern English Bulldogs cannot mate or give birth naturally. Their body proportions, massive heads, narrow hips, make natural reproduction impossible. Every Bulldog is born via cesarean section. Every pregnancy requires artificial insemination. But the real horror is respiratory function. Bulldogs' skulls have been bred so flat that their airways are severely compressed. The soft pellet, tissue at the back of the throat, is too long for the shortened skull. Nasal passages are narrowed or collapsed. The trachea is often malformed. Result? Bulldogs spend their entire lives in mild oxygen deprivation. They overheat easily because they can't pant effectively. They snore, gag, and struggle with even mild exercise. Lifespan averages 6 to 8 years, half that of similarly sized breeds. A 2016 study by the Royal Veterinary College, UK, examined CT scans of 50 bulldogs. Not a single dog had normal anatomy. Every individual showed severe airway compromise. The researchers concluded the extreme brachycephalic phenotype has reached a point where the welfare of affected dogs is significantly compromised. In the 1990s, neurologist Claire Rusbridge began investigating why so many Cavalier King Charles Spaniels screamed when touched or scratched at their necks compulsively, a condition called neck scratcher's disease. MRI scans revealed the cause. The skull was too small for the brain. This created pressure that forced brain tissue into the spinal cord opening, blocking cerebrospinal fluid flow. Fluid accumulated in cavities, syrinxes, within the spinal cord, causing excruciating nerve pain. By 2008, studies showed 95% of Cavaliers over age 5 have this condition to some degree. Many show no symptoms yet, but the structural problem exists in nearly the entire breed. The genetic cause? Extreme selection for an apple-domed skull, a rounded head shape specified in the breed standard. Breeders selected dogs with the most extreme skulls, inadvertently selecting for insufficient cranial volume. Golden Retrievers are America's third most popular breed, beloved family dogs known for gentle temperaments. They're also dying of cancer at catastrophic rates. A 2019 study following 3,044 Golden Retrievers over their lifetimes found that 65% died of cancer. By age 8, over half had developed cancer. The most common types, hemosarcoma and lymphoma, are highly aggressive and usually fatal. Researchers at Colorado State University traced the cause to genetic bottlenecks. The entire American Golden Retriever population descends from a handful of dogs imported in the 1930s to 1940s. Intensive line breeding to create show winners further narrowed the gene pool. Specific gene variants predisposing to cancer became fixed at high frequencies. With such limited genetic diversity, dogs lack immune system variation needed to fight cancer effectively. The breed has been bred into an immunological dead end. The numbers paint a devastating picture. Bulldogs, 80% develop breathing problems, 72% develop skin fold infections, 39% develop cherry eye. Boxers, 38% develop cancer, 42% develop heart disease. Dobermans, 58% develop dilated cardiomyopathy, fatal heart disease. Bernese Mountain Dogs, 50% die of cancer, median lifespan 7 to 8 years. Great Danes, 37% develop dilated cardiomyopathy, median lifespan 7 to 8 years. These aren't isolated problems affecting a few unlucky individuals. These are breed-defining characteristics affecting the majority of purebred dogs. Traditional explanations, bad luck, aging, environmental factors, fail to explain why these problems cluster so strongly by breed. A German Shepherd is 50 times more likely to develop hip dysplasia than a Greyhound. A Boxer is 20 times more likely to develop cancer than a mixed breed. The explanation is genetic. Intensive inbreeding has created populations carrying multiple recessive diseases at high frequencies, combined with immune dysfunction from lack of genetic diversity. But this represents just one piece of a much larger puzzle. If genetic diversity loss were limited to a few breeds or a few diseases, the problem would be manageable. But genomic analysis reveals something far more disturbing. The genetic crisis extends across the entire species. The most remarkable discovery involves what happened to dog genetics in the last 50 years. In the 1960s to 1980s, as dog showing became increasingly competitive and commercial, a practice emerged, using champion stud dogs to sire hundreds or thousands of offspring. The logic seemed sound. If a dog wins championships, it possesses desirable traits. Breeding it extensively spreads those traits through the population. But this created genetic disaster. A 2012 study by Imperial College London found that in many breeds, 5 to 10% of males sired 50% of puppies in subsequent generations. In extreme cases, single males fathered 20 to 30% of an entire breed. This amplifies genetic problems exponentially. If a champion dog carries a recessive disease mutation, and he likely carries several, that mutation spreads throughout the breed in a single generation. Example, an Irish setter named Chow Knockross Sable won dozens of championships in the 1960s. He sired over 220 recorded offspring. Today, genetic analysis shows he's an ancestor to over 50% of living Irish setters. He carried the mutation for progressive retinal atrophy, a blinding disease. PRA now affects 10 to 15% of Irish setters. This pattern repeats across breeds, champion dogs becoming disproportionate ancestors, inadvertently spreading genetic diseases that won't manifest until generations later. Breed standards specify ideal appearance in precise detail: skull proportions, ear set, tail carriage, coat texture, color patterns. Dogs are judged against these standards at shows. Winners become breeding stock. But here's the trap. Many breed standards specify anatomical features that are inherently unhealthy. German Shepherd breed standards changed in the 1970s and 1980s to favor a sloped back, with hindquarters lower than shoulders. This became fashionable in show rings. Breeders selected for increasingly extreme slopes. Result? Spinal deformity. The sloped back creates biomechanical stress on the spine and hips, contributing to hip dysplasia and degenerative myelopathy, progressive paralysis. Modern show-line German Shepherds often have such exaggerated slopes they walk with a distinctive frog-leg gait, hindquarters collapsing inward. This would be considered a severe structural fault in working-line German Shepherds bred for police or military work. The breed standard created a health crisis by rewarding extreme anatomy. In the 1990s, geneticists discovered something disturbing about Dalmatians. The genes for coat color are linked to genes for hearing. Dalmatian coat patterns result from a mutation affecting melanin production, but this same mutation affects melanocytes, pigment cells, in the inner ear, cells essential for hearing. Dogs with the most extreme white coats often lack functional melanocytes in the ear, causing congenital deafness. Result: 15 to 30% of Dalmatians are born deaf in one or both ears. Breeders selecting for perfect spotted coats inadvertently selected for deafness. This isn't unique to Dalmatians. Many breeds show genetic linkage between show-winning traits and health problems. Blue-eyed dogs often have higher rates of blindness. Extreme white coats associate with immune problems. Heavily wrinkled skin creates infection-prone fold dermatitis. Extremely long coats cause chronic eye irritation. Every organism carries some deleterious mutations. Geneticists call this genetic load. But in healthy populations, natural selection removes the worst mutations.
To understand how we created this crisis, we need to understand population genetics.
the mathematics of how genes flow through populations over time. A genetic bottleneck occurs when a large population is reduced to a small number of individuals. Those few survivors become ancestors to all future generations. Any genetic diversity not present in the survivors is lost forever. Dr. Heidi Parker at the National Institutes of Health reconstructed the genetic history of dog breeds using genomic archaeology, analyzing patterns of genetic variation to infer historical population sizes. Her findings revealed that nearly every modern breed went through severe bottlenecks. Phase 1, pre-1850s. Diverse working dog populations across regions, relatively high genetic diversity. Phase 2, 1850s to 1900. Breed clubs formed, breed standards established, registry closed, no new dogs admitted, population crashes to founder group of 10 to 100 dogs. Phase 3, 1900 to 1950. Intensive line breeding to fix breed characteristics, further diversity loss. Phase 4, 1950 to 2000. Popular Sire Effect amplifies specific lineages, additional bottlenecks. Phase 5, 2000 to present. Recognition that breeds are in genetic crisis, attempts to intervene. At each phase, genetic diversity dropped precipitously. Parker's calculations show modern breeds retain only 10 to 30% of the genetic diversity present in 1850. This isn't just academic, it's a genetic emergency. Conservation biologists working with endangered species would consider these numbers catastrophic, requiring immediate action to prevent extinction. This forces us to reconsider fundamental assumptions about what a dog breed is. Are breeds stable, genetically defined entities, or are they artificial constructs maintained only through constant human intervention? Populations that would collapse without cesarean sections, artificial insemination, and lifelong veterinary care. Population geneticists have long understood inbreeding depression, the fitness decline that occurs in inbred populations. Inbred organisms show reduced reproductive success, increased juvenile mortality, impaired immune function, reduced lifespan, accumulation of deleterious mutations. Wild populations experiencing inbreeding depression typically go extinct unless genetic diversity is restored through immigration of unrelated individuals. But dog breeds are closed populations. Breed standards prohibit outcrossing. The genetic damage cannot be repaired through natural mechanisms. It only accumulates. Dr. Adam Boyko at Cornell University proposes we're witnessing genetic meltdown, a feedback loop where inbreeding causes health problems, reducing reproductive success, forcing even more inbreeding to maintain populations, causing worse health problems. Some breeds may be beyond the point of recovery. The genetic diversity loss is so severe that even with perfect management, genetic load continues increasing. Not all geneticists agree the situation is hopeless. Dr. Jeffrey Kidd at University of Michigan argues sophisticated breeding strategies, carefully selecting mates to maximize genetic diversity while maintaining breed type, could stabilize breeds. His team developed algorithms that analyze pedigrees and genomes to identify optimal breeding pairs, combinations that would produce healthy offspring while conforming to breed standards. Early results show promise. Breeders using diversity-conscious breeding show slower rates of genetic diversity loss and modest health improvements. But critics argue this merely delays the inevitable. With effective population sizes of 10 to 50 individuals and closed gene pools, no breeding strategy can restore lost diversity. The damage is permanent without introducing new genetic material. Dr. Jessica Heckman at the Broad Institute poses a philosophical question that divides the dog world. Do we have an ethical obligation to preserve breeds as they currently exist? Breed preservation groups argue yes, breeds represent living heritage, centuries of cultural history. Allowing breeds to go extinct or dramatically change through outcrossing destroys that heritage. But animal welfare advocates argue no. When breed preservation requires maintaining populations in genetic crisis, causing widespread suffering, preservation becomes cruelty. Are we ethically justified in creating and maintaining populations we know will suffer simply because we value their appearance? This question has no easy answer. It forces us to confront fundamental values. Is breed purity more important than animal welfare? Is preventing suffering more important than preserving genetic lineages? The genetic crisis facing dogs isn't insurmountable. Emerging technologies offer unprecedented opportunities to restore health to purebred dogs if we're willing to make controversial decisions. Recent breakthroughs suggest we're entering a new era of canine genetics, where we can not only understand the damage, but potentially reverse it. In 2015, geneticists Adam and Ryan Boyko founded Embark Veterinary, a company offering direct-to-consumer dog DNA testing. For $200, owners can sequence their dog's genome and receive health reports. But Embark isn't just selling tests. They're building the world's largest canine genetic database, over 1.3 million dogs sequenced as of 2023. This database is revealing patterns invisible in smaller studies. Dr. Aaron Sams, Embark's chief science officer, explains, "We can identify disease-causing mutations at population scale, track their frequencies across breeds, and predict which dogs are carriers. This gives breeders unprecedented power to make informed decisions." Example: Embark identified a mutation causing degenerative myopathy, progressive paralysis, in Pembroke Welsh Corgis. By testing breeding stock, breeders can ensure they never produce affected puppies, even while using carriers. Within one generation, they could theoretically eliminate the disease from the breed. Embark has identified over 350 genetic health conditions, creating tests for each. For the first time, breeders have tools to actively reduce genetic disease burden. But the deeper revolution is diversity mapping. Embark can calculate each dog's genetic diversity score, measuring how inbred they are relative to their breed average. Breeders can use this to select the least related mates, maximizing offspring diversity. Early data shows promise. Breeders using Embark's diversity tools produce litters with 15 to 20% higher genetic diversity than breed averages. Some breeds have taken radical action, deliberate outcrossing, breeding to dogs outside the breed. The most successful example, Dalmatians. Remember, 15 to 30% of Dalmatians are deaf due to genes linked to coat pattern. Additionally, Dalmatians have a unique metabolism that causes them to produce uric acid stones, painful urinary blockages requiring surgery. In 1973, geneticist Robert Schappel bred a Dalmatian to an English pointer. The goal: introduce the normal uric acid metabolism gene. After 5 generations of back crossing to Dalmatians, breeding offspring back to purebred Dalmatians, the resulting dogs were genetically 96.9% Dalmatian, looked indistinguishable from purebreds, but had normal uric acid metabolism. The Kennel Club rejected them for 30 years. Finally, in 2011, facing mounting evidence of success, the AKC accepted them into the registry. Today, LUA Dalmatians, low uric acid, are healthier, live longer, and rarely develop stones. Some breeding programs have embraced them. But here's the controversial part. Outcross puppies are banned from most dog shows for multiple generations. This discourages breeders who depend on show wins for reputation and puppy sales. Similar outcross projects are underway for other breeds. Pug Breeding Project in UK, crossing pugs with Jack Russell Terriers to restore normal skull structure. Basenji Health Project in US, adding African village dogs to restore genetic diversity. Borzoi Outcross Project in Finland, adding Saluki bloodlines to combat genetic diversity loss. Results are promising. Outcross offspring show dramatically improved health while retaining breed characteristics, but acceptance remains controversial. The most radical possibility? Gene editing. CRISPR-Cas9 technology allows precise modification of DNA sequences. In theory, deleterious mutations could be corrected, restoring functional genes. Dr. Jennifer Doudna's lab at UC Berkeley demonstrated proof of concept in 2021. They used CRISPR to correct a mutation causing progressive retinal atrophy in dogs. Treated embryos developed normal vision. But ethical questions abound. Is it acceptable to edit dog genomes?
Who decides which edits are therapeutic versus cosmetic? Could this lead to designer dogs with enhanced traits? Or what about unintended consequences? Most geneticists urge extreme caution. Gene editing has potential to eliminate genetic diseases, but it also has potential for abuse. Regulations are needed before this technology becomes widespread. Perhaps the most fascinating possibility, ancient DNA from archaeological sites could restore lost genetic diversity. Dr. Gregor Larsson's team at Oxford University has sequenced DNA from dogs buried 5,000 to 10,000 years ago. These ancient dogs possess genetic variants absent from all modern breeds. In theory, these ancient alleles could be reintroduced. Techniques like somatic cell nuclear transfer, or cloning, or targeted gene insertion.
Let's follow the evidence to its most profound implications. The most profound implications emerge when we consider what the genetic crisis reveals about human responsibility towards species we've created. Dogs are the only species on Earth that humans designed from scratch. We took wolves and molded them into 400+ distinct forms, from Chihuahuas to Great Danes, through artificial selection. This makes us responsible for their welfare in ways that don't apply to wild species. We created their genetic problems. We perpetuated them through breed standards and closed registries. We prioritized aesthetics over health. Are we ethically obligated to fix what we broke? Some philosophers argue yes. Our intervention created suffering, therefore we must intervene to eliminate it. This might require abandoning breeds, accepting outcrossing, or implementing strict breeding regulations. Others argue intervention got us into this mess. Further intervention risks making things worse. Better to allow natural selection to operate by removing artificial breeding pressures. But dogs can't return to nature. They're completely dependent on humans. Natural selection can't operate on populations maintained through cesarean sections and artificial insemination. Inaction isn't neutral. It's a choice to perpetuate suffering. If we accept that some breeds are beyond genetic rescue, effective population sizes too small, genetic load too high, health problems too severe, should those breeds go extinct? This question divides dog lovers. Breed preservation advocates argue every breed represents irreplaceable genetic heritage. Letting breeds go extinct is a tragedy. But veterinarians treating bulldogs with collapsed tracheas, Cavaliers with brain-crushing skulls, and German shepherds with disintegrating hips increasingly ask, is extinction worse than suffering? If a breed cannot exist without chronic health problems, does it deserve to exist at all? Some propose modified preservation, outcross to restore health, even if this means breeds change appearance. Others propose genetic archives, preserving breed DNA and frozen cells without maintaining living populations that suffer. There are no easy answers. Perhaps the most fundamental question, should dog breeds continue to exist as closed genetic populations? Closed registries made sense in the 1800s when genetics wasn't understood. Now we know closed populations inevitably accumulate deleterious mutations and lose diversity. Maintaining closed registries guarantees genetic decline. Some geneticists propose opening all registries, allowing strategic outcrossing to restore health while maintaining breed characteristics. Dogs that look and behave like golden retrievers but are healthier because they carry 10% Labrador genes. Others propose eliminating breeds entirely, breeding for health, temperament, and function rather than appearance. Dogs optimized for their roles, family companions, service dogs, working dogs, rather than conformance to arbitrary aesthetic standards. The most radical proposal used genetic engineering to create super breeds, dogs with optimal health, longevity, and function designed from the ground up rather than through selection. Each approach has profound implications for the future of dogs and our relationship with them. Perhaps the deepest question, do we possess sufficient wisdom to redesign a species? Our track record suggests no.
We created the genetic crisis through overconfidence in our ability to improve dogs through selection. Now we propose to fix the crisis through genetic engineering, cloning, and advanced breeding strategies. What if we're wrong again? Geneticists urge humility. Evolution spent 40,000 years creating dogs through natural and artificial selection. Modern breeds have existed 150 years. We've barely begun to see the long-term consequences of our interventions. Perhaps the wisest course is admitting our limitations. Stop trying to perfect dogs and simply allow them to be healthy. Accept variation, value function over form, breed for welfare rather than winning. We stand at a crossroads. The genetic crisis facing dogs is solvable. We have the technology, knowledge, and resources. What we lack is consensus about what dogs should be. Should we preserve breeds as living museums, accepting health costs? Should we prioritize health, accepting breed changes? Should we redesign dogs using genetic engineering? Should we step back and let dogs become what they'll become without intensive selection? Whatever we choose, we cannot escape responsibility. We created dogs. Their suffering is our creation. Their future is our choice. The most profound question is whether we'll choose wisely.
We began with images of suffering: a bulldog struggling to breathe, a German shepherd's collapsing hips, a Cavalier's brain-crushing skull. The evidence has revealed something both devastating and hopeful. Devastating, through 150 years of intensive inbreeding, we've reduced many dog breeds to genetic catastrophes, populations more inbred than species on the edge of extinction, carrying multiple genetic diseases, suffering from immune dysfunction, living half the lifespans they should. We did this deliberately. We specified breed standards requiring unhealthy anatomy. We closed registries, preventing genetic rescue. We rewarded extreme traits at dog shows. We used popular sires excessively, spreading genetic diseases throughout breeds. We prioritized appearance over welfare. The suffering we see in purebred dogs is not natural. It's engineered. But hopeful, we now understand the problem. We possess tools to fix it: genetic testing, diversity mapping, outcrossing programs, and emerging technologies like gene therapy. We know what healthy dogs look like: ancient dogs, mixed breeds, working line dogs bred for function rather than form. We can restore health to dogs if we choose to. The scientific consensus is clear. Maintaining closed registries and breed standards requiring unhealthy anatomy is indefensible. Genetic diversity must be restored, through outcrossing if necessary, or breeds will continue declining. Some breeders have accepted this. Outcross projects are underway, breed clubs are revising standards to remove unhealthy requirements, Embark and similar companies are providing tools for diversity-conscious breeding. Progress is possible, but many resist. Show breeders whose reputations depend on producing dogs conforming to traditional standards, kennel clubs protecting century-old rules, puppy buyers who want dogs matching Instagram aesthetics. Change will require not just scientific solutions, but cultural transformation, recognizing that breed purity has a cost, and that cost is paid in canine suffering. Your dog, whether purebred or mixed, healthy or struggling, is part of this story. Every breeding decision, every puppy purchased, every breed standard vote contributes to whether dogs become healthier or sicker over coming generations. Dogs cannot advocate for themselves. They cannot choose their genetics. They depend entirely on us to make wise decisions about their future. The question is whether we'll prove worthy of that trust. Dogs have given us 40,000 years of companionship, loyalty, and love. They've adapted themselves to us in ways no other species has matched, becoming our protectors, workers, therapists, and friends. In return, we've reduced many of them to genetic ruins. We owe them better. We owe them health. We owe them the chance to live full lives free from chronic pain, respiratory distress, and genetic diseases. The science exists to make this possible. The technology is available. All that remains is will.
the collective will to prioritize welfare over aesthetics, health over heritage, dogs over dogma. The story of dogs isn't finished, and neither is our responsibility. The choice is ours. The consequences are theirs. Choose wisely.